A sealed vial of dust, collected off the most famous piece of cloth on Earth in 1978, was finally opened by a genetics lab and run through a next-generation DNA sequencer. Scientists expected either silence or a single signature. What came back instead was DNA from at least 14 different humans, spanning 3 continents, along with cats, dogs, cattle, carrots, wheat, fish, and insects that had no business being anywhere near a 2,000-year-old burial cloth. The machine had not found the fingerprint of one man. It had found the fingerprint of centuries. This is the story of what that DNA analysis actually revealed about the Shroud of Turin, why it reopened one of the oldest arguments in modern science, and why the real answer is stranger and more human than either side of the debate wanted. I am your name, and this is your channel. If real science with honest context is what you come here for, subscribe right now. We go this deep every week. Let us get into it. The Shroud of Turin is a 14-foot length of linen bearing the faint, brownish image of a man's front and back, along with what appear to be bloodstains consistent with wounds from flogging, a crown of thorns, and crucifixion. Its first confirmed appearance in the historical record is in France in the year 1354, when it was displayed at a small collegiate church in the town of Liray. It later passed to the House of Savoy and has resided in the Cathedral of St. John the Baptist in Turin, Italy since 1578. For believers, it is the actual burial cloth of Jesus Christ. For skeptics, it is an extraordinary medieval object whose true origin has nothing to do with the first century at all. In 1988, 3 independent laboratories in Oxford, Zurich, and Arizona performed radiocarbon dating on a small sample cut from the shroud's edge. All 3 arrived at strikingly similar results, dating the cloth to somewhere between 1260 and 1390. That window places its creation squarely in the medieval period, more than 1,000 years after the death of Jesus. For over 3 decades, this single test has functioned as the closest thing science has to a final verdict, and it is the reason most mainstream historians treat the shroud as a medieval artifact rather than an ancient relic. That verdict has never fully settled the matter because other researchers have pointed out real technical objections to it. The 1988 sample was cut from a single corner of the cloth, a section some conservators believe may have been patched or rewoven at some point during the shroud's documented history of fires and repairs. Critics of the radiocarbon result have long argued that a single small swatch taken from an edge that survived a major fire in 1532 and years of subsequent restoration work may not accurately represent the chemical composition of the cloth as a whole. In 2024, Italy's Institute of Crystallography published an analysis using a technique called wide-angle X-ray scattering, which measures the structural degradation of linen fibers over time rather than relying on carbon isotopes at all. That study produced results the researchers described as consistent, with the shroud being roughly 2,000 years old, directly at odds with the 1988 radiocarbon date. Neither side of this argument has produced evidence strong enough to fully silence the other, and that unresolved tension is exactly the environment the new DNA research walked into. It is worth pausing here, because this is the pattern that repeats through nearly every major shroud investigation of the last 5 decades. A team announces a new finding using a new instrument. That finding is technically real, peer-reviewed, and methodologically sound within the limits of what it measures. And yet, it still fails to close the argument because the shroud's history is long enough and its handling messy enough that almost any single measurement can be explained more than one way. The DNA study that follows does not break this pattern. If anything, it deepens it by adding an entirely new category of evidence to a debate that already had more open questions than settled ones. The genetic story begins with the same sampling event that produced the radiocarbon test. In 1978, during an extensive scientific examination of the shroud known as the STURP investigation, a researcher named Pierluigi Baima Bologna vacuumed dust particles directly off the cloth's surface, focusing on the areas corresponding to the face, the hands, the lower body, and the lateral edge later used for carbon dating. Those dust samples, containing pollen, skin cells, fibers, and microscopic organic debris, were preserved for decades. Bologna himself continued researching the shroud until his death in November 2025, and he remained part of the research team investigating those very samples until the end of his life. For years, smaller studies had already detected scattered traces of biological material in that dust. But those early efforts used older amplification techniques that could only search for genetic sequences researchers already suspected might be present, which meant the results were narrow and easy to dispute. The breakthrough came when an international team applied a completely different method to the same 1978 samples: PCR-free next-generation metagenomic sequencing. Unlike older techniques, this approach does not go looking for one specific type of DNA. It sequences every fragment of genetic material present in a sample simultaneously, then sorts the results afterward, producing a far more complete, picture of everything that has ever touched or contaminated the cloth. Published in the journal Scientific Reports, the results were, in the researcher's own description, the most comprehensive genomic picture of the shroud ever assembled. The sequencing identified diverse human mitochondrial lineages, skin-associated microbes, fungi, salt-loving archaea, and DNA from cultivated plants and domesticated animals. This was not a study looking for Jesus. It was a study cataloging everyone and everything that had ever left a genetic trace on a piece of fabric that had been touched, kissed, displayed, transported, and handled for at least 6.5 centuries, and possibly for 2,000 years. The human genetic results alone were remarkable. Researchers identified mitochondrial DNA sequences belonging to at least 14 separate individuals, clustering into a range of distinct genetic lineages built up over generations of contact with the cloth. Roughly 55% of that human genetic material traced back to lineages associated with the Near East, the region encompassing the Levant, the Arabian Peninsula, and surrounding areas, a result that at least broadly aligns with centuries of pilgrimage traffic from that part of the world toward European churches and relics. Close to 40% of the human DNA, however, traced back to individuals of Indian heritage, a finding nobody fully expected, and one that immediately complicated the story. Researchers also confirmed genetic material belonging to Boulogne, the scientist who collected the samples in 1978, a reminder that even the person studying the relic became part of its accumulated genetic record, and a small illustration of exactly how easily modern contamination enters historical evidence. None of these lineages appeared in isolation. They were layered on top of one another, overlapping in ways that suggest repeated contact over long stretches of time, rather than a single point of origin. A cloth sealed away for 2,000 years, untouched and unhandled, would not carry this kind of accumulated human genetic diversity. A cloth that spent centuries on public display, carried in processions and passed between religious institutions across 3 regions of the world, would carry exactly this kind of layered signature. The pattern itself, independent of any single lineage, tells its own story about how this object has actually lived through history. Now understand what this actually means before jumping to conclusions. A DNA sequence traced to Indian ancestry does not mean an Indian person was wrapped in this cloth 2,000 years ago. It means that at some point, someone with that genetic background touched, handled, wove, traded, or came into close contact with this fabric and left cellular material behind. Cloth passes through countless hands across centuries of trade routes, pilgrimages, restorations, and public exhibitions. Every one of those hands is a potential contributor to the genetic mixture scientists are now reading. The Indian genetic signature is not an isolated finding either. It echoes an earlier study published in 2015 by Gianni Barcaccia of the University of Padua, one of the researchers involved in the newer analysis as well. That 2015 study had already proposed, based on separate genetic evidence, that the linen itself may have originated in the Indian subcontinent rather than the Middle East or Europe. Seeing a similar Indian genetic signature reappear a decade later in an entirely different and more advanced sequencing effort gave that earlier hypothesis renewed weight, even though it remains far from proven. But that's not even the strangest part of what the DNA revealed. Alongside the human material, researchers cataloged an extensive menagerie of animal DNA embedded in the fibers of the cloth. The sequencing turned up genetic traces from cats, dogs, chickens, cows, goats, sheep, pigs, horses, deer, and rabbits. It did not stop there. The team also found DNA from multiple species of fish, including gray mullet and Atlantic cod, as well as marine crustaceans, flies, aphids, and arachnids. This is not the genetic profile of a burial shroud kept in isolation inside a stone tomb. This is the genetic profile of a textile that has spent centuries moving through markets, churches, storage rooms, and the hands of ordinary people who lived alongside farm animals and ate fish for dinner. The plant evidence tells a nearly identical story. A separate but related preprint, released in March, examined the same 1978 samples specifically for botanical DNA and identified genetic traces from carrots, bread wheat, and a range of other cultivated crops. The researchers behind that study concluded these plant traces suggest centuries of connection to a first-century burial. A related earlier peer-reviewed analysis had already found something similar. That study identified plant taxa native to the Mediterranean region, alongside species originally from Asia, the Middle East, and even the Americas, some of which were only introduced to Europe after the medieval period had already ended. A vegetable that did not exist in Europe until after 1500 has no plausible explanation on a first-century relic, unless that contamination happened centuries later through ordinary human contact. Read that again, slowly. Crops from the Americas cannot appear on a burial cloth from ancient Jerusalem unless that cloth picked up genetic material long after the age of exploration connected the two hemispheres. This single detail does more to anchor the shroud's recent handling history than almost any other piece of evidence in the entire genetic data set, and it lines up cleanly with centuries of documented European display and storage rather than an unbroken, undisturbed burial environment. Think about what daily life actually looked like for the people who guarded, displayed, and transported this cloth across 6 centuries of European history. Cathedral staff who ate meals near where relics were stored. Farmers and merchants who handled cloth in markets that also sold produce and livestock. Restorers who worked on the fabric using tools and materials that had touched countless other objects first. None of this requires any dramatic explanation. It requires only the ordinary, unavoidable mess of human life happening in close proximity to a piece of fabric for hundreds of years. Here is where the honest scientific caution has to come in, because not every part of this dataset carries equal weight. Christina Warrener, an anthropologist at Harvard University who studies ancient microbiomes but was not involved in the research, has publicly stated that the human and microbial results are likely trustworthy, given the sequencing method used. She has also cautioned that the plant and animal identifications relied on an alternative genetic matching approach, known to handle plant and animal sequences less reliably than it handles human ones. In other words, the presence of a scattered vegetable garden and a small zoo's worth of animal DNA on the shroud is very likely real contamination, but the exact species list deserves some healthy skepticism until it is independently confirmed using stricter methods. Now here is where it gets disturbing for anyone hoping this study would finally settle the age-old argument. It does not even close. Anders Götterström at Stockholm University, who was not involved in the new research, responded to the DNA findings by restating his confidence in the original 1988 radiocarbon dating, saying he still sees no reason to doubt that the shroud is French and dates to the 13th or 14th century. From his perspective, and from the perspective of many mainstream archaeologists, an ocean of contamination DNA changes nothing about the fundamental chronology already established by physical dating methods. Genetic diversity proves the cloth has been extensively handled. It does not on its own prove when the cloth was made or who, if anyone, it originally covered. If this kind of evidence-first investigation is what you come here for, hit subscribe and turn on notifications, because we take apart claims like this every week, following the data wherever it actually leads rather than wherever the headlines want it to go. The genetic findings arrived alongside another study that reshaped the conversation, just as significantly, though it involved no DNA at all. Using 3-dimensional modeling techniques, researchers compared the body image on the shroud to the proportions and shadow patterns you would expect from an actual human corpse lying flat versus the patterns produced by a low-relief carved sculpture draped in cloth. The 2025 study concluded that the image formation was more consistent with a medieval sculptural technique than with actual contact against a human body. If accurate, this suggests the famous image itself may have been produced using an artistic method available to medieval craftsmen, rather than any biological or miraculous process acting on real human remains. Taken together, the genetic study and the sculptural modeling study point in a similar direction, even though they used completely different methods and different research teams working independently of one another. One suggests the cloth has absorbed centuries of ordinary human, animal, and plant contact, consistent with long-term circulation through medieval and early modern Europe. The other suggests the image on its surface behaves more like an artistic reproduction than a natural imprint from a real body. Neither claim is proven beyond dispute, your research using better instruments than the ones available today. But neither claim supports the idea of an undisturbed, sealed first-century relic either, and that convergence, arrived at through entirely separate lines of evidence, is harder to dismiss than either finding would be on its own. So what did the DNA actually reveal, stripped of speculation? It did not reveal the genetic code of Jesus of Nazareth. No scientist involved in this research has claimed that it did, and no serious geneticist believes a 2,000-year-old body, if one were ever present, could realistically leave dominant, recoverable DNA after nearly 7 centuries of documented public handling, fires, floods, and repeated restorations. What the sequencing revealed instead is a layered archive of nearly every group of people, animals, and crops the shroud has come into contact with since it entered the historical record, spanning the Near East, Europe, and South Asia, stitched together across roughly 650 years of continuous human contact. That is not a disappointing result. It is, if anything, a more interesting one. A single cloth carrying the accumulated genetic residue of pilgrims, priests, restorers, traders, farm animals, market food, and crops from 3 continents is a working historical instrument in its own right, regardless of what anyone ultimately concludes about its religious origin. It documents in molecular detail centuries of exactly the kind of movement, veneration, and handling that written historical records already describe happening to the shroud after 1354. The debate over the shroud of Turin's true origin is not resolved by this research, and it likely will not be resolved by any single study in the near future. The radiocarbon dating points to the medieval period. The 2024 crystallography analysis points toward extreme antiquity. The DNA study points toward centuries of accumulated human, animal, and plant contact. The sculptural modeling points toward artistic technique rather than a literal body imprint. Every method looks at a different layer of evidence, and every method has genuine documented limitations that its own researchers openly acknowledge. What remains constant through every single one of these studies, regardless of which side of the debate they seem to support, is that the shroud itself keeps generating real, testable, peer-reviewed science almost 50 years after the first modern investigation began in 1978. Few objects on Earth have been measured, sequenced, X-rayed, modeled, and argued over with this much technical rigor for this many consecutive decades, and the fact that scientists are still examining genetic evidence that did not even exist as a research tool when this investigation began tells you the story is far from finished. Every decade brings a new instrument capable of reading something the previous generation could not access, and each one adds detail without ever fully closing the case in either direction. That is unusual for an object this thoroughly studied. Most historical artifacts, once dated and cataloged, settle into a fairly stable consensus that later research only refines at the margins. The shroud has resisted that pattern for half a century, not because the science around it is weak, but because it carries so many independent, testable layers of evidence, chemical, genetic, and visual, that each new method reveals something the last one could not. Go back to that sealed vial of dust, opened after nearly half a century in storage. Nobody who unsealed it found the DNA of one man from the first century. They found the accumulated genetic fingerprints of 14 different people, a small farmyard of animals, a market's worth of vegetables, and centuries of ordinary human contact, all preserved in the fibers of the most argued over piece of cloth in history. The real revelation was never going to be a single answer written in genetic code. It was always going to be the sheer, staggering volume of human history the cloth had quietly absorbed while everyone kept asking it the same unresolved question. Drop a comment below with which finding in this video surprised you most, the Indian genetic lineage, the American crops, or the sculptural modeling study, and share this with someone who still thinks this argument was settled back in 1988.